Hello, and welcome to today's webinar titled Enhancing Cell Line Development, Viability and IgG Detection with Cyto-Cellect. I'm Colette Cooley, and I'm delighted to be your host today on behalf of Sphere Bio. At Sphere Bio, we're committed to driving innovation in single-cell analysis and biopharmaceutical research, and today's webinar is an exciting opportunity to share some key insights with you. But before we dive in, a few housekeeping notes. The webinar will last approximately 45 minutes, and if you have any questions, feel free to put them into the chat box and we'll address them at the end. A recording of this session will be available afterwards, so don't worry if you miss anything. Now it's my pleasure to hand over to our speaker, Ben Douglas. Ben is one of our senior application scientists from the US. Ben, over to you.
Thank you so much for the kind introduction, Letty. Good day, everyone. My name is Ben Douglas. I'm a senior field application scientist here at Sphere Bio. I'm incredibly excited to be your host this afternoon as we discuss how Sphere Bio can enhance cell line development workflows with our newly launched Cyto-Cellect Viability Kit. As a brief preamble, I'm going to provide a quick overview of our company, our current product offerings and solutions for those working in antibody discovery and cell line development settings, to paint a complete picture before diving into the specifics of the Cyto-Cellect and Viability Kit. So who are we? Sphere Bio is a company based in Cambridge, UK, with fully staffed direct sales teams in North America and Europe, as well as extensive distribution partners in the Asia-Pacific region that aim to deliver best-in-class microfluidic-based solutions for antibody discovery and cell line development workflows. At the time of this webinar, we have over 300 customers globally and an IP portfolio consisting of 135 patents and counting. We're continuously evolving to ensure that we continue to deliver these best-in-class solutions to aid our current and prospective customers in removing common pain points that are present in conventional and antiquated workflows. So what are these pain points? Early-stage biological discovery and development requires extensive screening efforts to identify and select the top-performing cells. Standard practices typically include starting from large heterogeneous populations or mini-pools and drilling down to select a desired number of clones that tick all of the necessary requirements to bring those selected forward in the development process. In a standard cell line development workflow, these initial steps historically have been conducted by utilizing limiting dilution or FACS to isolate cells into wells of microtiter plates. The clonality of each well is then confirmed by eye using microscopy or a plate imager, and then the waiting game begins. Typically over the next 1 to 2 weeks, outgrowth and production kinetics are observed to start parsing down the desired hits that are then moved on to the next stage in the development cycle. Not only are these methods slow and have insufficient levels of throughput, but they often require major investments in capital, personnel, equipment, and importantly, time. To mitigate these pain points, we have developed a series of microfluidic instruments and reagents to condense the selection process of early development timelines from weeks to a few hours. Our latest instrument release is the new generation of Cytomine, the Cytomine Chroma. The Chroma was officially launched at SLAS earlier this year, retains the ease of use and speed of the flagship Cytomine instrument, but now houses an updated optics module that is now equipped with 4 lasers, 4 detectors, enabling a true multiplex approach to the cell selection and screening, and expanding the breadth of functional assays that can be run on the system to help you find the cells you want faster. The Cytomine Chroma combines 3 core technologies together to create an approachable and intuitive workflow that can be executed by anyone in the lab, no matter what skill level. The beating heart of the Cytomine Chroma is the ability to encapsulate cells and reagents into picodroplets. This is carried out by forming a single layer emulsion of the aqueous and reagent suspension in our patented oil and surfactants. These droplets are 450 picoliters in volume and approximately 100 microns in diameter, allowing for single or multicellular encapsulation of antibody-producing cells. This small volume allows for the rapid buildup of these secreted antibodies that can be accurately measured by the Cytomine Chroma. These droplets are gas permeable and highly stable, facilitating for optimal cell health for the duration of the experimental run. These picodroplets serve as the reaction vessels for the functional assays that can be run with the chroma. To get a sense of the scale we're working at, a standard well of a 96 well plate filled with 100 microliters is the equivalent of 100 million picoliters in volume, which is a 200,000-fold difference compared to our 450 picoliter picodroplet. This reduction in volume again allows for the buildup of secreted antibodies from the single cells, which can be detected in as little as 1 hour, compared to the conventional microwell approach, which can take days to weeks to generate an appropriate level of product to detect by conventional means. The core functional assay that is utilized by the chroma is the FRET assay, in which fluorescently conjugated probes that bind to specific regions of the secreted antibody aid in the detection of that product. When both probes are bound to the antibody within 10 microns of each other and subjected to fluorescent activation, the donor probe will transfer energy to the acceptor probe that will indicate a shift in fluorescence. This generates a diffuse signal inside the picodroplet that is recognized by the integrated detectors to zero in on the droplets that contain cells that are active in producing the antibody of interest. The final core piece of technology is the focal point of the entire operation, the cytocartridge. All the necessary functions performed on the chroma all take place in this one location. That's droplet generation, droplet sorting, dispensing, and imaging all occur in this prefabricated off-the-shelf consumable. The only intervention needed by the end user is loading the cell suspension into the cartridge and loading the cartridge onto the manifold of the chroma, and then the system takes care of the rest. So putting all the core technologies together gives us an ultra simple and refined process for early selection in the cell line development workflow. This starts with the heterogeneous cell suspension being hyperdiluted to a concentration of 500,000 cells per mil in 1 milliliter of encapsulation media, along with assay kit reagents and support supplements. This is then loaded into the cytocartridge, and then the cytocartridge is then loaded onto the cytomine chroma. Picodroplet generation then occurs, utilizing gentle pressure to generate the emulsion of the aqueous suspension and the oil layer. The droplets are initially kept at a stable 4 degrees C until all the droplets are generated. The user can either generate 1 million, 1.5 million, or 2 million picodroplets, depending on their throughput needs. The droplets are then shuttled to the incubation chamber through a series of microfluidic channels embedded on the cytocartridge, where the temperature will then rise to the desired setting, allowing for an optimal environment for antibody production to occur. The incubation period timeframe is completely customizable by the end user that can range anywhere from 1 minute to 24 hours. Once the incubation period is complete, the droplets are then cooled back down to 4 degrees prior to sorting. The droplets are then sorted using dielectrophoresis and by gating on the desired droplet population that exhibit a positive FRET signal. Any non-sorted droplets are diverted into the waste chamber. These non-sorted droplets are typically droplets that contain non-secreting cells or those that are void of any cellular material altogether. The sorted droplets are then transferred to the dispensing chamber and again sorted on the desired populations of interest based on the FRET signal. Here is where the end user can select the high, medium, or low producers to dispense. Each droplet is imaged 5 times at different stages in the dispense chamber to serve as an evidence of monoclonality of the droplet. The droplets are then dispensed into pre-filled microtiter plates, either 96 or 34 well formats, at a frequency of 1 Hertz or roughly 1 well per second. From this point forward, standard practices for monitoring outgrowth and titer can be utilized. So to quickly summarize, the cytomine chroma can effortlessly slot into the existing suites of tools needed for cell characterization and selection in the early stages of development. Coupled with our off-the-shelf reagent kits, prefabricated consumables, intuitive software packages, and an overall ease of operation, the cytomine chroma offers a robust entry into microfluidic workflows that enables our customers to increase their throughput, reduce their consumable costs, but more importantly, focus their attention on their scientific needs rather than the needs of a complicated piece of equipment. So now on to the main topic of today, the Cyto-Cellect Viability Kit. This assay kit is an expansion of our established Cyto-Cellect kit, which includes 2 distinct types of probes for functional testing: human IgG Kappa probes and viability probes. The human IgG Kappa probes use the described FRET-based mechanism. Donor and acceptor probes bind specifically to the human IgG secreted by cells contained within the picodroplets. When both FRET probes are bound, energy transfer occurs from the donor showing green fluorescence to the acceptor showing orange fluorescence. The shift in signal indicates the presence of secreted human IgG inside the picodroplet. Second, the viability probes consist of an Annexin V conjugated with a far red fluorophore. These probes are utilized to detect picodroplets that contain cells undergoing apoptosis. As the cells are dying, the phosphatidylserine present in the inner leaflet of the cell membrane flips to the outer structure of the cell membrane. Annexin V binds to this exposed marker, and the far red signal allows us to identify these dying cells. The combination of these probes allows the end user to drill down on the picodroplets that are not only actively secreting antibodies, but those that are also healthy and non-apoptotic. So next, I briefly just wanted to walk through the performance data of the assay kit. Figure A on the left represents the ability of the FRET probes to detect the main classes of IgG Kappa at varying concentrations. This POC experiment was performed in a plate-based assay prior to running on the cytomine chroma. Figure B shows the standard sorting plot seen on the cytomine, showing the fluorescent shift in detection of the droplets containing actively producing cells, and those that are either empty or not producing. These are represented by the positive gate located closer to the y-axis, representing the orange signal of the acceptor probe, and the negative gate nearest the x-axis, representing the green signal of the donor probe. The combination of these two distinct probes again allows for the simultaneous assessment of productivity and viability. This is achieved on the chroma by using a common method in FACS, serial gating. Figure A shows a scatter plot of cells during the sorting process, illustrating the distribution between picodroplets that contain a positive signal for FRET and a positive signal for the Annexin V viability probe, signaling apoptotic cells. If we further drill into these populations of both the far red negative and far red positive populations, we can observe that both of these populations are producing antibodies that can be detected using the FRET probes. This highlights the importance of including a viability dye when performing a functional IgG secretion assay. Using the information from the combination of these two probes, we can sort on the droplets that are negative for the Annexin V dye and positive for the FRET signal. This allows the end user to enrich the droplets for dispensing that enables the best chance for cell survival. Figures D and E on the right show the viability and total cell counts after 13 days of culturing the sorted populations. Cells that were Annexin V negative during the sort demonstrate increased viability in overall cell numbers compared to those cells that were Annexin V positive during the sorting process on the chroma. So the Cyto-Cellect viability kit fits nicely into our existing portfolio, which we're continuously developing new assays in the area of antibody discovery, cell line development, and cell therapy. If you'd like more information regarding any of our assay kits, please follow the QR code to our website located in the bottom right corner of the slide. Thank you so much for joining us today. I'd be more than happy to field any questions that might have come up during this presentation. Thanks again.
Thank you, Ben, for that insightful presentation. We've received some great questions from our audience, so let's dive into the Q&A. The first question I have is, how long does it take to carry out one experiment with cytomine chroma?
Yeah, so it depends on a few factors. So the first being what type of application we want to do, right? So either antibody discovery or cell line development. So that require a few different inputs. The main topics being the number of droplets that you want to generate, the overall hit rate of the expressing antibodies that you're looking at, and then also the incubation time that the end user can set. So all those coupled together would ultimately result in about a 4 to 7 hour workflow. But the one key feature is that really the cytomine chroma takes care of all of the heavy lifting. So really the end user just has to load the sample into the cartridge and then pop the cartridge onto the chroma. And then the cytomine does all the rest. So even though it seems like a long day, but ultimately the overall hands-on time is actually quite minimal.
Thank you. And the next question, can we re-incubate if the expression level isn't high enough?
Yeah, so the system will allow the end user to look at the overall FRET expression, and after the desired incubation time, say if we incubate for 1 hour, we don't see the desired FRET level that we were expecting, we have the ability to go back and re-incubate the droplets that were generated in the incubation chamber for a longer period of time. And then from there, we can resort those droplets and redispense the droplets as well.
The next question. Sec. Okay, so can you compare this to a beacon and cell selector at other competing instruments that do similar processes?
Sure. So I can't really comment to the specific nature of each instrument, but as far as overall throughput, the cytomine does offer an increased level of throughput depending on the workflow. So you have the ability to screen, again, depending on the application. If we're talking antibody discovery, we have the ability to potentially screen 40 million cells over the course of one experimental run. Or if we're doing a CLD type of a workflow, we dilute it down to about 100,000 cells to encapsulate. Unfortunately, like I said, I don't have extensive knowledge as to how those other systems operate, so I can't really quite comment fully on their mechanisms of action as far as viability screening goes.
I think if we do have any other experts in the company that do know the answer to that question, we will get back to the person that asked that question. So thank you for asking that, actually. And your answer actually covered another question, which was how many cells can you load into the cytomine chroma? So that's perfect. That's all we have. Questions. Okay, that was a short one. Thank you very much, Ben. That brings us to the end of today's webinar. Big thank you to you, Ben, and for your expertise and your time. If you'd like to ask a question at any point that you remember after the webinar, feel free to reach out to us. Our email address is contact@spherebio.com. And we hope you found this webinar interesting, and we look forward to seeing you at other Sphere Bio events in future. Thank you very much.
Thank you.